Why, hello, my little coffee bean. Welcome to the Caffeine Club.
Hello, welcome to the stream. How are you guys doing? Happy Tuesday. I hope everyone's week is going well. And yeah, let's see. We have Laughing Underwater here. Laughing Underwater, thank you so much for being first tonight. Welcome in. Nyahala, you are third. How are you doing? I hope you're well. Sarmaleth, thank you so much for that sub. I appreciate it. I really do. And also, fun fact, guys, every time we reach 20 subs, I do a coffee giveaway. So we are 2 subs away. So, just throwing that out there, not trying to sell anything. But yeah, it is a Tuesday. I am a little bit frazzled. It's been a long day. But I am here. We're going to be doing kind of a short but not too short stream today. It's going to be, wow, that's like right in my face. Thank you. Thank you, laughing. I appreciate that. This is going to be the one stream I probably do for this week, because I'm going to be out of town this coming weekend. But there is a potential that I might do a vacation stream again, but we will see. The whole point of vacation is to get away from everything. But I really want to do a Bripe stream. And if you guys are new here, a Bripe is a very novel device used to brew coffee in. It's very interesting, but it looks sketchy, so you can't just Bripe anywhere. So that's why I was thinking, oh, maybe I'll just take it with me and do a little Bripe stream in the woods where no one will see me doing that. But anyways, who knows? Brandon Halcyon, welcome in. Hello. You are going to work and cannot watch long. Well, I hope you have a good time at work. You're going to work right now. Oh, you're going to work early in the morning. That's true. Okay. Well then, I hope the rest of your week goes well. You have a crazy work week. Let's just give it up to all the people who have to do 6-hour or six day work weeks. That's ridiculous. It's a lot. It's a lot. But yeah, today I got my very first undergrad in lab, and I'm very excited. So basically, one of the high points of being a grad student is at some point you get a young little minion that learns to become like you, and you get to take care of them and teach them how to do things. And in the process, you realize how much you've learned along the way. And so for me, I was I was a little surprised at some of the things that we had to go over, but overall it was pretty fun. And today we were doing something where we basically purify DNA from bacteria. And there's actually a very fun video I'll show you guys in a little bit, but it's basically what you do if you want to make a lot of a certain type of plasmid, which is basically a little circle piece of DNA. You use bacteria to grow. Basically, the bacteria will, as they replicate, they'll also replicate the DNA of your choice inside of them. And then at the end, you lyse them and take out the DNA and purify it. So that's what we were doing. And I think it was a fun experience. Also, today I wanted to go over some AMA questions that were left in my Excel sheet. So if you're not familiar, you can also just go to this little link here. I don't know if it's working. It's a very ugly link. I'm sorry. I need to fix that. But what I have kind of started doing a little bit is to give you guys a chance to ask your various questions. Hang on, trying to find the right Excel sheet. What? It's signed out? Okay, hang on. There we go. To give you guys a chance to kind of ask questions related to cancer or immunology, I thought it would be better if people put their questions in a comment form in an Excel sheet, so that way I have some time to like think about the question and give it a thoughtful response, because otherwise, you know, it's like asking me something on the fly usually works, but sometimes if it's a very deep question, I need a little bit of time to come up with a good answer and find good sources. So let me see if I can share this. Where's my window capture? Hang on. Here we go. So this is currently the sheet. There are a number of questions that I have been DM'd that I need to add in here. But basically, my categories that you can ask me questions about are the immune system, microbes, or cancer. And one of the questions that I was going to go over today is actually not on the sheet, but it's relating to chimeras. And the question was mostly about the ethics of chimeras, which are basically a chimera is a combination of two different species. And this could include let's say, a human host or human cells that are placed inside of, say, a mouse or a primate. If the primate, for example, is the host and you're putting in human cells, for example, then you would say that's a chimera. And usually the goal of these chimeras is to grow an organ or something that kind of contains a mix of both of these two different species cells. There's also hybrids, which we aren't going to get into, but hybrids is where you would have the egg and then the gamete from two different species. So you would get a mix of genetic material. And the user who was interested in this, I don't think he's here right now, but it was Styles Este, and he was concerned about the ethics of making chimeric humans. And also there was this very interesting story that I will just show you guys. There was a very, I guess, kind of jazzed up article written about chimeras, and I thought it was a little sensationalistic. But I'm just going to share it with you guys right now. Okay. So as you can see, first of all, worst musical ever. I haven't watched it, but I don't want to watch it. Oops, why is it showing this? My bad. Hang on. That should not be happening. What the heck? Give me a second. We have some chaos happening. Patwick! Thank you for that. Make me mostly caffeinated. Give me 1 second. Hang on a second. I messed up my scenes. Why is it? Dude, oh my god, why is this even like this? Okay, there we go. It should not have been screen capture. I don't know why that was on. That was, I think, from another time. Very weird. All right. So we can do a mostly caffeinated. But first of all, before we get there, I guess we could start with a caffeinated thing. So a little coffee making. If my camera turns on, if it does not turn on, then we will not do that. But what I want to get into tonight is this fun little article. Hang on, camera is trying to turn on. Things are just going chaotically. I reset my computer by accident, and so all my settings kind of just went straight out the window. And, you know, things aren't lined up properly, like this window page. There we go. So the sensationalistic article is basically suggesting that, so first of all, Japan did not approve a plan to specifically make cat girls. That is, I know it looks like that, but that is definitely not what's going on here. And human animals is also an incorrect term. Basically what they're allowing is letting them use chimeras in research or studying and making chimeras, and only up until a certain amount of time. So not letting these chimeras become born, so only going up to 14 days after fertilization, and then cutting it off, because there are a lot of ethical concerns in terms of bringing chimeric humans or animals into the world that we have not addressed yet. But that is going to be one of the articles we go over. And then there was also another article on this topic that was very interesting from Stanford, but that is on my other window, so we're going to just open that later. But right now, Patwick has redeemed a slightly caffeinated, and so that is something we do on this channel, is we make coffee. And so, you really caught me off guard, dude. I was planning not to get caffeinated. I was literally drinking a downer, trying to relax, and then you come in here and you caffeinate me. I cannot believe you. All right, we're gonna make a pour over because it's very simple and I don't have all my espresso things out right now. You're good. Maybe I will save some of the leftover coffee for later. Hang on, let me just put a little text box up real fast so that people know what's going on, because it is a little bit weird to come in here and see coffee making happening. So give me 1 second. Let's add a new source. There we go. All right. There. Let's see. Where's the text? Wait, where did it go? Are you serious? Oh, it's so small. Whoa, it's so big. Okay, wow. It's very blurry. The resolution is not great. Okay, that is fine. That's fine. All right. There we go. Okay, so we are going to be making some coffee very briefly. So the coffee that we're going to be drinking today is, I know, the same keynote. I've been drinking this all week, and it's something, if you work in a lab, for example, you know that caffeine is essential. And so I've mostly been making pour-overs because it's the quickest caffeine beverage to make. Damn it, Patwick, I don't even have water in my pot. Okay, I will be right back.
Not.
What the heck? Dude, I missed it. I only heard the last part. Jay, welcome to the stream. How are you, Jay? We are making some coffee briefly because Patwick decided that I should not sleep. So that is what we're doing. Okay, so most people who are coffee snobs, they actually weigh their coffee. But if you're in a rush and you don't want to do that, you can just estimate by pouring in a similar amount of coffee that you do every morning. That is what I do when I'm in a rush. Oh my god, laughing. I just saw your comments. So we have some coffee grounds. I use the Baratza Encore. It's a pretty affordable grinder if you're, living on a budget, such as myself. It was about $100 for a pretty good grinder. This isn't a blade grinder, it's a burr grinder. So if you're trying to up your coffee game with a more even and consistent grind size, I think it's a pretty good grinder. I know there's a lot out there, and I'm not particularly attached to it, but I would say it's pretty good. So we're waiting for our water to come to about 90 degrees Celsius. And while that's happening, I'm going to grab my Chemex. So the Chemex is basically this gigantic funnel thing, and you can make coffee in it very quickly. So yeah. I feel like it kind of looks like a flower vase. If you didn't know any better, you might just gift it to somebody with a bouquet in it and call it good. It's like one solid hefty piece of glass. Now, I have a tendency to break things, and I've only had this for a month, so we're gonna just knock on wood and hope that I don't drop it at some point, because it does feel very breakable. It's pretty thick, but it's also, again, I'm sure if you dropped it hard enough, it would just shatter all over the place. So I like to use, so the filter paper that you use for this is actually not this. This is a Hario filter paper, which is designed for a much smaller V60. The Chemex filter paper is much larger, and it actually comes in a square, and you're supposed to fold it into a 3-layered filter. Now, I think that's a lot of paper, and I personally don't like to do that. So I'm using up my old Hario V60 filters for the Chemex, even though they don't fit perfectly. As you can see, it keeps wanting to pop out. That's fine. We're going to put some water in it, and it'll be just perfect. Now, some people like to pre-wet their filters, but I've done a very moderate amount of testing where I have tasted the water that has gone through just filter paper alone, and it doesn't taste very different from just normal water, so I don't think it's affecting the taste of my brew too much. I know that's not like a great test. It's a very subjective test, but it works. So our water's heated. So I have put the coffee in. As you can see, it's somewhat evenly spread on the bottom, which is great. And we're just gonna go ahead and pour some in. So usually I like to pour roughly about 50 grams in. And then I just kind of let the coffee bloom. And I know it's a little bit hard to see, but we're getting some blooming going on here. And this is just to release some of the gases that are trapped in the grounds from the coffee. Only 50 grams. Yeah, it's a very small amount of water, but we're going to add more water, don't worry. I usually add 350 grams. And if I'm going to be honest, I'm guesstimating right now because as you can see, I have no scale. This is a very highly scientific process. It's called winging it. And it's something I do all the time, which, really gives everything I do flair. You have no idea what you're talking about, but also add Milo. Maybe I will add some Milo. That's just more caffeine and sugar. I'll add some Milo. I have some in my drawer. Okay, so we finished the bloom. So the next part is kind of fun. You can't really see it, so I'm going to tilt it a little bit. You just pour a little bit of hot water just straight on the middle. And then we're going to slowly make little circles around. You can kind of see the water now as it's filling up. Now, if this was on a scale with a timer, I could theoretically measure flow rate. Now, you see something interesting going on here. So what's happening is the filter paper has formed sort of a vacuum, and so as the air that's being pushed out, or basically as the hot water is flowing down, there's air that's being pushed up, and it's causing this bubbling action to happen. And you can just remedy that by burning yourself like that, or you could just remove the filter paper and create a little air gap so that it doesn't bubble so much. Fit for Games, thank you so much for that follow. Welcome to the stream. I hope you're doing well. I hope you like coffee. We're currently on a coffee break, but we're going to be getting back to some more science-related stuff in a second. I know I'm still on that. But yeah, hope you're doing well. So sometimes I like to stir the edges to kind of knock the grounds that are sticking off, but we're not going to do that right now. I think most of the coffee has settled, which is nice. Don't add Milo. Oh, you're giving me mixed signals, dude. Milo is good in basically any sort of caffeinated beverage, so yeah, I wouldn't have not tried it. All right, so honestly, we're gonna finish with one more little pour, and then I think that's enough because I didn't do too many grounds. I did roughly 20 grams, not 20 grounds, but 20 grams of coffee. And this is roughly 150 grams of water, maybe more like 200 at this point. But what I do sometimes is I'll dilute my coffee, and it's very disgusting. Don't do it. You can always add water. You can't take it back. So try to start off with a more concentrated coffee, so less water, and then add it to the cup if you're feeling that it's a little strong. Which, honestly, if you think it's too strong, then you're kind of a wuss, but whatever. one more pour. You're paid by the liter. Yeah, no. I'm sorry. Water's limited here. It's California, okay? You have to pay for your water. You have to pay for your coffee water. Okay. So I think that's good. I'm going to go ahead and pour the coffee in a mug, and yeah. Okay. So you see it's settled. There's no pitting, which means that the water flowed through evenly. Usually you have to stir to kind of make sure it doesn't do any pitting, but we didn't have that problem today. Probably because Patwick is a little good luck charm. All right, so you can squeeze your little bag out, and then you can fertilize things with your grounds, or you could just throw them in the trash. I don't have plants because I kill them, so I'm gonna just throw this in the trash. Okay, so now you can see we have a lovely brew. And I'm just gonna pour this into my mug that I didn't wash from this morning because I was in a rush. Okay, so today I decided to go to the gym before lab, and I also had to get to lab by a specific amount of time. And so I was very late, but I still made time to brew some coffee, but I just didn't clean anything afterwards, which is kind of always disgusting because then you get like those little coffee stains inside of your stuff. So that is what I had going on. All right, so here it is. This is our coffee made with the Keynote medium roast beans. They're delicious. Highly recommend. They've been okay for espresso. I haven't really enjoyed them as much in an espresso shop by themselves, but they're okay for pour overs and things like that. Only medium roast. I know we didn't do like dark roast or light roast this time. Light roast is very tangy. I haven't been liking the light roast that much. It's actually watery. I think I added too much water. That's fine. That's fine. Okay, guys. Going back to things. Let's turn this very bright light off. So going back to the little Chimera paper. Let's see. You only deal with maximum roast? It's maximum roast. Is this when you put it in like a French fry thing and just heat the oil up max all the way? That's gross. You just keep roasting until your beans are a char, and then you just take them out of the roaster and powder them in your fingers and call it good. Yeah. So, okay. Let's go through this paper. So, this paper was, or this article is based on a real scientific paper, but this article was published in 2019. And Stiles brought up the question. He was like, are they making chimera cat girls in Japan? And from this very sensational image, one would think that, but that is not the case. So basically, there was a group that, here's the Nature link. This is the original kind of Nature news article, so not the research paper, but a news article that was discussing how this Japanese research group was trying to gain support from the Japanese government to allow them to pursue human-animal embryo experiments. And so what this means is that they would inject human cells into animal embryos and then they would put those embryos back into a pregnant or pseudo-pregnant animal host, and the animal host would carry the embryos to 14 days, and then they would terminate the pregnancy. Now, what's the whole point of this? The point of this was to actually create organs, and these would be human organs that could be used in like human transplants, for example, because one of the big problems that we have right now, I'm sure you guys are aware that there is a shortage for organ donors, and so people will just never get the organ that they need, and they'll pass away while they're waiting for an organ transplant. And so this research is not geared towards making human-animal chimeras for shits and giggles. The real goal of the research is to basically use an animal host as a biological incubator for making new organs that can be transplanted into humans. But the organs have to be made out of human cells, otherwise the human body will reject that organ. And so that is the whole point of this very sensationalistic sounding research. Gear, hello Gear, welcome to the stream. How are you? Hang on, let me just turn this. I feel like I'm very much lopsided today. My desk has been a little bit chaotic lately. Anyways, Gear, how's it going? How's your week? I know it's only Tuesday. Hopefully it's still good. That's from 2019. Yes, this paper is from 2019. And actually the original Nature article or the Nature News article talking about this was published July 26th, 2019. So, like 4 years ago. So it's been kind of a thing that's been happening. But before you get excited, let's get rid of this picture because it's very misleading. So for the first time, the reason this was so big is there is no current government in any country, any regulated country, that supports human animal like chimera research due to different ethical concerns. So this was the first time that a government decided to support a plan to create animal embryos with human cells and bring them to term. So actually they're kind of pushing the 14-day limit, resulting in a type of human animal known as the human animal chimera. According to Nature, a committee from Japan's science ministry signed off on a request by researchers to grow human pancreases in either rats or mice, the first such experiment to gain approval since a government ban was reversed. So they're starting small. And what I heard about this study, which we'll see later in another paper, is that a lot of these experiments kind of result in failure because the cells don't actually grow into like the desired organ that they want them to. Oh, you're curious about the results. Yes. Okay. So in this paper, or in this article, let's see here. I don't know. Oh my God, look at all these personalized ads. I'm so sorry. I'm obviously a peasant who can't afford to get rid of ads. But basically, okay, so they created these embryos. So before this even happened, researchers had also been creating human animal embryos with sheep and pig embryos in which they added human cells. I think this is the paper for that. This is another article written by the same guy. Okay. Yeah, this is actually from 2017, and he's talking about using the pig human embryos to grow and harvest human organs. And so let's see. I think they experimented with several different combinations: rat cells injected into mouse embryos, rat cells injected into pig embryos, human cells injected into pig embryos. The rat-pig chimeras did not work at all. So one thing that has to be considered too is how evolutionarily related the two species are that you're trying to use to create a chimera. Obviously, rat and pig are somewhat further skewed than, say, a non-human primate and a human. Let's see. What is this? So last year, so in 2016, they reported that the scientists who were working with the human cells, they were injecting human cells or blank cells. So basically, these are a type of stem cell into 1,500 developing pigs. So developing, I want to say that those are pig embryos, not like actual full-on baby pigs. So 1,500 pig embryos when they were, then they clarify, when they were just proto-embryo balls of blastocysts. So what does that even look like? Let's look at a picture of a blastocyst so you guys aren't imagining a small pig in your head, because that would just be wrong. So a blastocyst is literally a little clump of cells. Doesn't look like much, but it has a lot of potential to develop into a whole-on like animal or human. So this is what they're injecting human cells into. So they take a blastocyst from a pig, they put human cells into it. That's the primary thing. Sloth. Oh my god, sloth. What if we made a sloth chimera? I think that would be so exciting. We could make a sloth human chimera and we could name it, I don't know, Fluffy. The man bear pig. What is the man bear pig? Is this an internet sensation? Can I link you the follow-up article from this year? Oh, is it from the same guy? Jayski, you can send it to me. Okay, so they're right now they're like still talking about the stuff that was happening in 2016 when this was kind of new. Let's ignore the ad. So the ultimate goal is to generate cells for human tissue. All right. So why didn't the human pig chimera work out as well as the rat mouse variety? So here they go. So basically the human pig chimera, spoiler, didn't really make the organs they were hoping for. But the rat and mouse chimeras did. And that's because rats and mice are much closer on the evolutionary tree than humans and pigs. That's one reason. Also, humans and pig embryos take different amounts of time to grow, so getting the chimera to thrive takes tedious coordination. So I guess this would relate sort of to size and size of organs, because organs from a human are going to grow to a much larger size, although they are similar to a pig. So I think they thought that maybe the timing would be all right, but there was still a big enough difference where it was challenging to get the human organ to grow properly inside of the very small pig. And then picking the right stem cells could also determine how much human tissue thrives. So I think one of the biggest updates is that they have found a better stem cell to use, and they seem to be having more progress now with that. Okay. None of this means, okay, blah, blah, blah. Okay, yeah. If a pig-grown organ future makes you queasy, you're not alone. So this idea, I didn't know this, but it's been around since the 70s. And obviously, it's a very big point of contention. A lot of issues, Okay, I have a question for you guys. How many of you, if you were, lying on your deathbed and you needed an organ transplant, would you take an organ that had been grown in a pig? If it was, presumably told, like obviously a lot of testing had been done, a lot of clinical trials, would you be likely to take that? Oh, you sent the Discord. Perfect. Thank you. Oh wait. Oh, okay. This is this paper is, this is the one that I saw earlier, I think. Or, no, maybe it's one similar with a similar name. Okay, I will take a look at this. Thank you. Yeah, this is all about the new Japanese regulation on human chimeras or human animal chimeras. Okay. Oops. Damn it. Okay, going back to this. Okay. One of the big questions is, what if a human brain cell grows inside of a pig's brain and you get this sort of weird hybrid where you have a pig brain and a human brain kind of combined? And there was a study where in mice, they took human brain cells, glial cells, which are responsible for a lot of memory and cognition, mostly memory, I think. They call them the thinking cells. They put these human glial cells into developing rats or mice, and the mice with the human stem cell or glial cells were they said they were smarter. Now this is based off of different types of tests that they have for mice, but they were basically, their conclusion was, okay, the human cells did something to change the mouse cells so that their brains were a higher functioning than their normal mouse counterparts, which is a little interesting and a little strange. And definitely, I would say cause for concern if you're going to go ahead and, try to create a human-animal hybrid brain. You're going into some pretty messy territory, I think. Lovely Lou, you redeemed a hydrate. Perfect. I have some grape juice here. It used to be water, so we can drink this. It sounds like it's being blocked ethically, though. From Japan? I think they're trying to constrict the length of time in which you can allow a human chimera embryo to grow within its host. I heard that the limit was 14 days. I don't know if that's what the paper is saying as well, or if they reduced it even further. Okay, so that's this older thing. This is the newer paper, but not really that new. Let me pull up the other article that's actually from 2022. That is not what I want. There's a Stanford Medicine article from 2022 that discusses current research in this area. So let me just put that there. Okay. All right. So in this paper, this was written in August 30th, 2022. It's an article just kind of summarizing a bunch of different research papers that have come out in the last few years. And let's see here. Basically, okay, this is again discussing about how all of this chimera research is stemming from the desire to find a new way to make organs. Some people, like I'm sure you guys have heard, I don't actually know, but have you guys heard of organoids? So organoids are basically petri grown organs. And so they try to grow 3D organs in a cell culture dish. Normally, things that grow in a cell culture dish are kind of constrained to growing very like flat or in a monolayer, as we call it. But there are ways where you can kind of stimulate the cells to grow into potential organs, and people mostly want to use those in research as kind of an alternative to animal models because, if you want to do proper like in vivo research, you normally have to use some sort of animal model like a mouse or something. But because that's not ideal, like people wish they didn't have to do that, we wanted to see if we could use organoids instead. But that's still going. I think there's some progress, but we aren't using organoids anytime soon. That's all I can say. Okay, so I see a question from Jay. The best question raised was that if they create something autonomous, can it consent to being created? I saw this question. It is also, what about the embryo cells from humans? Imagine that you're a female, for example, and you are donating your embryonic cells for research, right? And they create a chimera human animal cross hybrid thing with your embryonic cells. Are you the parent? Is it now your child? How do you approach that? It's a very weird, potential, very futuristic question, but also not too far in the future if people actually didn't have regulations and actually proceeded with this kind of research. Anyways, that's a great question. McScruff, hello, welcome in, McScruff. How are you doing? We're talking about chimeras today. I think you probably heard about the time when Styles was like, oh, they're making cat girls in Japan. That is not true. But they're trying to make human or primate or pig or other combination chimeras to grow organs in so that we can use those organs for transplants. But there are a lot of concerns because that kind of research is obviously stepping over some boundaries. Hoju, hello, welcome in. Is this where I come for the finest pour in all of Twitch? Well, yes, we have some coffee, but we also have some wine. There's a lot of different pours going on. I always have to kind of mix my upper with my downer, I hope you're doing well. I saw that you joined the Discord. I'm so happy. Yes, you are the biological parent, but should not be subjected to social economic responsibility. Yes. If you're signing over that biological clump of cells to a research facility, but then what if that chimera has some level of consciousness? Then what do you do? Okay, these are very theoretical or hypothetical questions, but definitely interesting. So going back to this, let's zoom in a little bit. So let's see, where was I? Okay, so because the pig human chimera wasn't working out, basically they would inject human cells into a pig embryo, but it didn't really develop the organs they were hoping for. Okay, and there's also a lot more restrictions in the US than Japan. But now the same team, so I think it's this team from Michael Snyder's group, but I could be wrong. Multiple people are studying this. I have no idea where this link is going. It looks sketchy. I'm just going to forget it. But basically, one research team has turned away from using pig cells to using non-human primates as sort of this biological incubator. We are using chimpanzee cells in this study. Okay, so in the US, you aren't allowed to use human embryos in research. So here they're taking two somewhat similar but rather distinctly unrelated species and then using that as sort of a proxy to combining animal cells with human cells. So here they're doing a study where they're using chimpanzee cells, which are genetically similar to humans, and they're seeing if they're trying to show that the chimpanzee stem cells can survive in a different animal model, in this case another type of monkey called a rhesus macaque. And then they're hoping that the viability will let them continue exploring this for actually making full-on functioning organs. Actually, let's see where this link goes. So this is a researcher named Hiromitsu Nakauchi. And in his group, he's a stem cell and genetics professor. Does he have his own lab? Let's see here. This is a very boring, research. He's currently studying different types of cells and organs and induced pluripotent stem cells. Okay, it is not clear to me he has recent publications. Very interesting. I suppose I should look more into his work at some point, but we will continue doing that. If you guys are interested in this article and all of the links that come with it, here you go. Oh, did the shout out not work? Here. We can do the shout outs in a little break. That sounds like a topic for Jerry Springer or Maury Povich. You are the responsible father, and the crowd goes wild. Who is Jerry Springer and Maury Povich? I've never heard of these entities before. Are they like... I've heard of the Jerry Springer show, and I thought it was like a very weird show where people would discuss weird things, but I don't actually know. If the chimera has consciousness when you hand it over. Wait. Oh, what is the question? If the chimera has consciousness when you hand it over. Well, if it was a small blastocyst, I don't think it would have consciousness, but that is obviously a huge topic of debate. But that's a good point. Jandre Loon, welcome in. Oh, the shout out worked. Hello. Daytime talk show host that started in the 90s. Oh, okay. I think there's a Weird Al song that discusses the Jerry Springer show, so if that makes you feel any better, yeah. You guys know the Weird Al song where he's like something about the Jerry Springer show, and it's like a very fast, weird song. You feel old. Both are old talk show hosts on Maori. Often he had people come in for paternity tests, and his popular line was, "You are the father." Oh my gosh. Wait, so how was it that he happened to have the actual fathers there? I guess, okay. They were hoping they were the actual father, and then sometimes they were not the actual father. Yeah, that is actually interesting because there are some cases, and this is kind of going into a realm of genetics I'm not familiar with, but there are cases where some people have tested negative for their paternity test, so their child didn't seem to be theirs, but there was basically other proof to confirm that that was indeed their child, and it turned out that the child was a natural chimera. And how that exactly works, I don't know. It's a very rare scenario, and there's only a few published cases of it happening, but that is an event where your child might not actually test positive, or you might not have a match in DNA if you do a paternity test. So that's always a possible but extremely rare possibility. Anyways, fun random fact for you. Okay, so let's see. Were there rhesus macaque and chimpanzee models successful? No. So multiple research teams tried this approach, but none had been successful. But then there was a Stanford veterinary research scientist named Morteza Rudgar who developed a new method to generate more stable and robust chimpanzee pluripotent stem cells. Now, stem cells are these cells that basically have the capability to turn into almost any cell in the body. And that's based off of what kinds of growth factors and signals you give them. So like different protein treatment like signals, because they have different receptors, and then that will lead to basically this like internal cell decision to become, let's say, like a muscle cell or a bone cell or a skin cell or something. So pluripotent stem cells, very versatile, but they are extremely hard to grow. I have some friends who work with stem cells, and they complain to me all the time about how they have to go into lab every day to take care of their freaking stem cells. They're so finicky. I grow cancer cells, and those things, you can like leave them for a few days and they'll be fine. But with stem cells, you have to feed them certain nutrients and different growth factors so that they keep proliferating properly. Okay, so basically this person, Morteza, was able to finally culture these really difficult chimpanzee stem cells. And so then they published a paper, which I believe hopefully is this link. It's called Chimpanzee and Pig-tailed Macaque Induced Pluripotent Stem Cells, Improved Culturing and Generation of Primate Cross-Species Embryos. So here it seems like their final, basically they're able to co-culture chimp, and human pluripotent stem cells. They cultured those together in a plate and then differentiated them into heart cells or heart muscle cells. That's interesting. What else did they do? This is just like the highlights. The whole paper is actually open access, so you guys can read it if you would like. Did they actually get let's see, what's this picture of? This is a very poor resolution picture. So it looks like they designed a protocol where they injected the chimp. So they took the chimp's like little stem cells, grew them up in culture, and then injected them into a rhesus embryo. And it says low iPSC survival. So I don't know how well this actually worked. Usually they always post the more positive sides of papers' results, even if it didn't work perfectly. But here you go. Oh my gosh, was sperm not allowed? Ugh. Guys, it's a biological term. We can discuss that. Some hoped, some didn't. The excuses were funny. I have low sperm count. It was someone else's, or there would be multiple possible dads wanting to know who is the father. That sounds like a really fun reality TV show. I love really horrible TV shows. Sometimes. Nick Saga is another edgy streamer that studies microbiology with a focus on DNA. Ooh, okay, I'm taking their name and I'm inputting it into my search history bar for later. Thank you for that. You're currently watching me in a VR movie theater. So in your little VR movie theater, can you throw popcorn at the screen? Is that something you can do? Okay, I will look for this person. Scientist, gaming enthusiast, nerd, book lover. Oh my gosh, wait, she sounds like my best friend. All right, moving on. Oops, wrong screen. Okay. So there's this paper. You guys can look at it. I'm not going to go through it at the moment. Probably. I'll leave it up just in case. All right, there's this guy. We don't care. So they were able to culture the cells. They published their paper in August 30th of 2022. Okay, so now scientists have been chasing the prospect of generating human organs, blah, blah, blah. Okay, there was another interesting part here. Nakauchi. Oh, so the guy whose profile I pulled up, he is a pioneer and longtime proponent of interspecies organ generation. So basically mixing the egg and egg and gamete donors to generate sort of a hybrid, I believe. I'm not sure exactly. There's so many definitions for interspecies organ generation, so I'm not entirely sure what they mean here. It could mean just a simple mix of cells from two different organisms, or two different species, or it could mean actual crossing of their genetic material from two different species, which are two completely different things. In his early work, he demonstrated that it's possible to create a rat pancreas in a mouse. Okay, this is like that 2016 paper. I think he must be the same guy, maybe. Or maybe multiple groups did that. His team showed that the organs generated inside the body of another species can be transplanted back to the original species without rejection. So this is the important crux of this whole thing. So normally with organ transplant, the person who is receiving the new organ has to be put on immunosuppressants because your immune system will generally attack any sort of tissue that is not matched based off of its genetic sort of makeup, but also, most importantly, its MHC makeup. So basically all over the cells of a tissue, there are these different proteins, and they are unique to each person. And some people have more similar kind of arrays of these proteins, and other people, their proteins are vastly different. And if they're vastly different, the donor or the recipient will reject the donor's organ. And so here they're trying to sort of match the organ with its potential host, so that way it's not rejected. So that's really important. Because usually if you're put on immunosuppressants, you're more susceptible to getting other illnesses and stuff like that. You threw popcorn at me in your VR chat? Oh my gosh, dude. You watch Paleontologist. He liked my dad jokes, so I followed. Okay, I have seen, I also follow Paleontologist, paleontologizing, or something like that. Basically, he's a streamer who discusses dinosaurs, and actually I saw that he was doing some live streams out in nature where he was doing actual digs, which is really cool. Paleontologizing. I always say that wrong. Thank you, Jandra. Jandre, sorry, I always say your name wrong, and I apologize. Fantastic! Welcome to the stream. I hope you're doing well. We are talking about chimeras and their use in making organs that we can potentially use for organ donations, and stuff like that. Okay. He's out in a dig and will be there 1 to 2 months. He has a Starlink set up to get net to stream with. Oh man. Oh, he's got the bougie internet. That's okay. I was wondering how he got such great service out in the middle of nowhere. I'll be honest, I only popped by for a little bit. So I didn't know what his setup was, but that is a lot of commitment. Yeah, he's very cool. You guys should check out Paleontologizing. I'm sorry, it's been a long day. Okay. So going back to this. All right. So let's see. Humans and large animals. Okay. So primate and rodent biology is very different. So basically, I don't think they've been able to necessarily replicate their rodent results in rats and mice in primates. Okay, I talk about the difficulty in culturing primate cells, stem cells. And then there was a bit of a pause in the field. This generally happens sometimes if things are difficult. But with this new technique from Morteza Rudgar, they are able to now continue this research in primates, which again are more genetically similar to humans. The biggest win was the viability of the cells that the team demonstrated with non-human primate cells in an early embryo, which was more akin to a clump of cells than anything resembling a living organ or animal. So they're still trying to get to the point where they can actually get a real organ to actually develop. Right now, this is more of kind of a proof of concept. Can their cells from the other species, can they actually survive in another animal's embryo. Okay. So here they're just looking at viability. So they said 2 days later, the implanted cells were still alive. That's it? Did they do longer? We were able to show that the cells survive, replicate, and grow. Okay. Well, they're going to need them to grow a little longer than 2 days, but these things take time. Getting cells to live in a whole other organism for 2 days is actually pretty impressive. Small step, but, a step in the right direction. So still proof of concept. Yeah, and then the ethics, which of course are, a bit of a hurdle sometimes, but also you want to make sure that you're not crossing any boundaries. So yeah, let's see. I think one of the biggest things and one of the biggest concerns is making sure that the stem cells that you implant, like human stem cells, if you were in other countries that allow that, you want to make sure that those don't actually develop into some sort of brain or nervous system in an animal. So anyways, very interesting paper or article, and there are links to the actual research papers if you are interested. Otherwise, the TLDR is that we are still making progress in actually getting primate stem cells to basically take and grow in their other primate hosts, but there is some progress. There is a group of edu-streamers called the Knowledge Fellowship. Wait, is this related to Science Stream? I think I might have joined this Discord group. I have so many Discord groups open in my Discord, but I might have joined this Knowledge Fellowship Discord, but then it fell into the pile of different servers that I have going on. I'll have to look for it later. Yeah, I don't see it. But I do watch Science Stream. I think that was the first edu-streamer that I kind of bumped into. And, I thought it was very fun, high energy place to hang out. You follow over 100 edu-streamers. Have you heard of Phaseria? She is more of a music streamer, but sometimes she does microscopy, which is very fun. I think it's more of a hobby, but she has like this fun sort of microbe identification bingo that you can play, and that's really cool. And I recently bumped into a streamer, and I'm forgetting her name, but she basically will draw different medical illustrations on stream and then discuss, I guess, whatever it is that she's studying. So she's like a medical student. Yeah, I don't remember her name right now, but anyways, okay. Oh, so let's see here. I just wanted to see if there was some other tab I had not explored yet. I have a bunch open at the moment. Yeah, this is a very hefty paper and not something I have read yet, so I don't want to try to cold read it right now. But yeah, I'll leave that there. And that one I'm going to plan to read later. Also, if you guys have questions relating to immunology, microbiology, or cancer that you would like as a potential discussion topic on a stream, you can add it as a comment to my little Excel sheet. And I will try to include it in a stream. I do have a lot going on in my normal life. I do research full time, but I do also like to kind of delve outside of my field and kind of see, what are things that people find interesting, and then kind of explore things like chimeras, for example. I would never even think about chimeras if it hadn't been brought to my attention by someone else. But one thing that I kind of wanted, okay, so last stream I was trying to hook up my iPad, and I think that it finally worked this time. So I'm going to try to hook it up right now again. Let me see. I think it's on my game capture. I was gonna kind of give a little diagram of how organ rejection happens, since I think it's somewhat relevant to what we were discussing earlier. Does this? Nope. Hang on. I just need to reactivate it. Is it active? No, I think it timed out. Did it time out? I honestly sometimes, so there's these like mirroring apps and they like to just time out after a specific amount of time, which is always fun. Okay, let's see. Tap record. Is it working? Jandra, I'm really curious. What is your specific interest in I guess, like, what is your, are you just interested in all different fields of science, or do you like have a specific interest? Okay, I'm just trying to get my iPad connected. I think it's, oh, yes, here we go. Yeah, it looks terrible. Let me flip it and make it larger. I don't know why it, oh, I know why it shrunk, because you have to maximize this window, which is super annoying. There we go. I think that's a little bit better. Still off center. Yeah. And we'll just zoom in like that. You've heard of TKF from Prof. Melko. Oh my gosh, TKF sounds so familiar. Who is TKF? Prof. Melko. Is this? So I know, okay, I don't think it's related, but there was a chemistry professor who liked to do his office hours on Twitch I think it was over the pandemic is when he started it. And then he just started streaming other things too, because it turned out that people were really interested. But Prof. Melko, who was that? That sounds so familiar. Yes, it is this guy, I think. Or is it a guy or is it a girl? Let's see. It is the chemistry guy. Okay, yeah, this is the person I'm thinking of. I followed him, but then I realized that I don't think he streams anymore. Or I just missed his streams. I'm not sure what happened there. All right, tell me if you can see my pen. That is a terrible color. All part of my autism is just learning. That is honestly really fun. I feel like learning is a constant process and really useful. He worked in the Senate for a bit. Oh, I didn't know that. What was his role in the Senate? Was he like a scientific advisor? Also, Starblam, welcome to the stream. How are you? I hope you're doing well. Yeah, I thought it was very interesting. I always think it's very cool when professors and other people in science start utilizing a platform like Twitch, for example, to kind of reach their audience a little better. I think that's very inspiring and fun. And that was kind of the reason why I decided to switch a little bit away from, so I mostly do coffee making and gaming on my channel, but lately I've been trying to transition more into science and technology, because that's basically what my 90% of my life revolves around. And so I thought it was a little bit silly of me to just try to kind of keep splitting myself 3 ways, which I still do because, you know, I'm insane. But yeah, I think, I don't know, I'm just trying it out. Science and technology, am I about to learn how to print 3D or 3D print coffee? Ghost Flame, welcome in. No, we were talking about chimeras and basically using a kind of a human and also maybe like a non-human primate or another animal like a pig, and basically creating, so like you would take a pig embryo and then you would inject human cells into that embryo to grow an organ. And basically you would want the organ to be a functional organ that you could transplant back into a human, and you're using the animal host as sort of an incubator. Now, this is a very controversial topic, and currently in the U.S., we're not allowed to do that with human embryonic cells. But most of this research is happening with, it was happening with rats and pigs, and then it was happening with different species of primates, since they're more genetically related to humans than pigs. But currently, the TLDR is that no successful organs have been made yet, but progress is slowly happening, as is often the case. He was a scientific, he had to step away for a bit because he got a job in DC as an advisor. He's back in Florida now and getting back to regular streams. Oh, awesome. Okay. He has a newborn child that eats up free time. Oh, that is really rough. That's a lot to handle. Some of my friends who have newborns, they don't have a lot of time, and I know it's very, very difficult. Scientific advisor to Marquis. Okay. Interesting. Space Turkey. Hello there. Welcome to the stream. How are you doing? Okay. So basically, one of the reasons why we are so interested in being able to generate organs that are basically either from a very similar genetic background as ourselves. So ideally, in the future, it would be nice to have sort of more genetically matched organ donors or donations. And the primary reason for this is on your cells. So let's say that we're going to just draw an organ. I'm so sorry, my drawing is atrocious. But let's say you have an organ. I don't know, this could be maybe a liver or something, and it's composed of all these different cells, right? And so let's just do a little zoom in. Let's say you zoom in on a part of the organ, and it's composed of cells, and all of your cells in your body, they have these molecules on them called major histocompatibility complex 1 or 2. That's actually for mice. On humans, we call them HLA. So you've probably heard of like HLA-A or B or C. And so on a normal cell, which again, terrible drawing, I know. Let's say you have an MHC1 molecule. So it has one part of the molecule goes into the cell, and then the other part is this tiny little subunit right here. And we're just going to call this MHC1 because, again, I work mostly with mice, so that's what I'm used to calling it. On humans, it would be like an HLA and then whatever variation it happens to be for that particular person. And then on this little MHC1 molecule, you have a peptide that's presented, and a peptide is just basically a little string of amino acids. This is kind of a zoom in, and it is basically originating from kind of junk proteins and also other proteins that are made within your cell. So there's a whole process, but basically your body will chew up leftover proteins, and then they'll get loaded onto an MHC1 molecule, and then through some little vesicle trafficking, they'll get stuck out onto the surface of your cell. And so basically the whole point of this MHC1 peptide process is to be able to label all of the cells in your body with whatever is going on inside of them. So let's say that, this cell becomes infected with some sort of virus, so there's a little virus inside the cell. What will happen is that your body's different enzymes and proteasomes will chew up the virus, and then you'll get these little viral pieces of protein that'll get chewed up, and they might get put onto MHC1. So then if you have your MHC1 molecule here, and you have a little viral peptide on it, your immune cells, like a T cell specifically, and we're going to get rid of this. This is kind of silly. Let's say you have a T cell. T cells have a special receptor. So T cells are able to recognize your MHC1 peptide complexes on your cells. And your T cells are kind of surveying your body to look for foreign proteins or viral proteins, for example, or cancer proteins. And so here's a little T cell. And then it has a little receptor on it, and this receptor will recognize this peptide MHC complex as a foreign complex. So basically, the T cell receptor will bind strongly to this little virus peptide and then the MHC complex, and it will tell the T cell to kill this cell. Now, normally, let's say this is just like a normal protein, right? So let's erase the little virus peptide, and let's replace it with the pink peptide, which is like, let's say, a normal protein from inside the cell. Sometimes when your body is making proteins, it messes up, and so those messed up proteins get recycled, and then they get loaded onto your MHC1 molecules, and those get presented on the surface. So, where's my pink? Okay. If this is just a, like a normal peptide, the T cell receptor will not recognize that. There won't be strong binding, and then the T cell will just kind of go away and ignore this cell and leave it alone, and it won't kill it. Oh, you're sleepy, Ghost Flame. All right, good night. Have a good one. By the way, I don't know if this is going to actually happen, but I'm going to try to do like an outdoor stream on Saturday, depending on my circumstances. I'm going to like this wooded area, but there's going to be a lake, and presumably there will be internet service to some extent, and if so, I plan to do a coffee brew pipe stream. If you guys don't know, you can brew coffee in a pipe. It's a very unique thing. It's not a real pipe. It's like literally a little brew pot for coffee. But yeah, that'll hopefully be Saturday. We'll see. I'm excited. I always try to do one bribe stream each year, which honestly, I've only done it once, so we'll see. But anyways, have a good rest of your week, Ghost Flame. Gustard, welcome in. How are you? Thank you. Also, Space Turkey, thank you for that. Is this your job or what you study, or both? This is my, I guess, job and what I study all combined into one. So right now, the reason I don't stream so much is because I'm currently in a graduate program for immunology, and my research is focused on how the immune system recognizes and responds to cancer. And also a little sort of offshoot of that is I'm interested in retargeting the immune system against the cancer, so like immunotherapy. And so one of the sort of kind of byproducts of that is I have to know how the immune system recognizes different cells in the body, and this is one such mechanism, this MHC1 peptide presentation. There's also MHC2, which presents peptides to your CD4 cells. So just for those of you who are immunology nerds, I need to be correct and say that this is a CD8 positive T cell. All right. You actually have multiple different types of T cells in your body, but the main two categories that often come up are CD4s and CD8s. And CD4 cells, they are these sort of little fun cells that, I shouldn't say fun, they're mostly known for providing help to CD8 T cells by producing cytokines, which are these little protein sort of messengers that sort of make CD8 T cells better killers. So you can imagine a CD4 T cell as being sort of like a cheerleader by producing cytokines. So let's say you have your CD4 T cell. It has its own little T cell receptor. So we'll make it look slightly different. And this CD4 T cell receptor recognizes MHC2, which is, it's basically, it looks like this, but with another kind of transmembrane sort of domain. And when the CD4 T cell gets activated, it will produce this cytokine called IL-2. And then IL-2 will be accepted by the CD8 T cell on a little IL-2 receptor. And it's made out of 3 different subunits, and that'll become more important if we ever talk about different IL-2 therapies. So actually, IL-2 therapy has been kind of explored as an anti-cancer therapy for almost probably 20 or 30 years, basically because IL-2 makes these CD8 T cells kill cancer better. So let's just say better killing. So this is good if we're trying to fight off cancer cells. And so basically people have tried to inject IL-2 just straight into a human, for example. First they did it in mice, of course, and the results were fine, like the cancers went away. So for example, let's say you have a little mouse, and they would give the mouse a tumor of some kind, and so this is the tumor, and then they would inject in, let's do this color, IL-2, which again is just like a purified protein version of it, and then the tumor would basically shrink. And so they were like, okay, this seems to be working, let's try it in humans. Well, guess what happens when you inject IL-2 into humans? You activate all of your T cells and you create sort of this like almost autoimmune sort of response where you get a lot of like cytokines that are produced and a lot of inflammation and things just go crazy. And a lot of people got very sick. I'm not sure, but I think someone actually died from getting an IL-2 injection. But I do need to look up that fact, but I think that was way back in the day, when they were not necessarily regulating these types of clinical trials that well. Free the nukes. What kind of link is that? If you're trying to send a link, you can, just because I have it so that people can't do links, you can post it in the Discord and I will look for it there. Laura Willingham, welcome in. Define and identify immune system and how you believe there's an actual system. So an immune system, I guess you could think of it as sort of like a cooperation. I guess which could also then be defined as a system because there's a lot of inherent organization that's going on. So every cell within the immune system, there's many different types of cells. They all coordinate and signal to each other, and this results in a specific type of immune response against a bacteria or against a cancer cell. And so that's kind of, and it's also something that is pervasive throughout the body. So immune cells in, like, let's say the brain can basically signal to immune cells in one of your lymph nodes just by the drainage and sort of, but there's like these like sort of, I don't want to say drainage systems, but basically, let's say there's certain types of like cancer proteins that are from a brain tumor. They can basically pass through and go into different lymph nodes, and then they can cause an immune response to those cells in the lymph nodes. But yeah, I don't know if that answered your question actually. Basically, I like to think of the immune system as a gigantic collection of cells with all their own unique function, and they're all very interlinked with each other. And if you change one thing about one immune cell, you're most likely going to change the whole system to some extent, and so you have to be aware of that. Free the nukes. Oh, brew pipe. Oh, did you look it up? I have one of those. They're so niche. There's actually a Reddit, a subreddit just for brew pipes, and it's really funny. Basically, people will go on there and they'll take pictures of themselves like brewing coffee in a pipe, and then they'll post it. Do your studies also include AI? So not yet. I would say I'm in a very traditional immunology lab. So actually we don't use AI for anything in our lab currently. But some people who are more focused on using a lot of sequencing data. So for example, let's say you have a tumor, for example, and let's say you have a tumor section. Sometimes people will try to predict what kinds of peptides different tumor cells might, or they'll try to predict what kind of T cell receptors would bind to certain types of tumor peptides. That's not really AI though. That's more like a predictive algorithm, which I guess is kind of AI. I don't know. It's been around before the whole ChatGPT thing blew up, so it's not necessarily like that. But I know that I do not do any sort of fancy programming or anything. Yeah. Oh, so what I was going to explain. So sometimes what people do if they get like a cancer biopsy from a patient and they want to sort of figure out different traits of that cancer so that they can better treat that specific type of cancer. Let's say like, I don't know, a melanoma or something. I can't spell melanoma. So what they'll do is you can use sort of like a different types of antibodies, for example, to stain different proteins that might be expressed on the cell. You can also do this in a more high throughput way where you use basically, I guess I should just stick to this one example. Let's say some different types of cancers will escape the immune system by losing expression. Remember, this MHC1 molecule, it's required for the CD8 T cell to recognize whether or not a cell is good or bad. Well, a cancer cell can just completely erase this, and basically they get a mutation in a specific component of the MHC1 molecule, and so the cancer cell no longer expresses that. So let's just shift over here. So a cancer cell will sometimes lose MHC1. And so then you won't have any recognition by the T cell. So here's your little T cell. Now it's blind. It cannot see it. The name tag has been removed. No MHC1. No killing. This is sort of the central accepted dogma right now, I guess you could say, or belief. However, there are some exceptions to this. So what happens is sometimes you'll get these cancers, you treat them, you give them an immunotherapy to make their T cells better, but then you'll get a subset of these cancer cells that lose MHC1, and so you'll get a tumor that kind of, let's say these are all MHC1 negative cells. Basically, they'll all grow out, and there's maybe a small subset of like the MHC1 positive that are remaining, and those will get killed off by the T cells, and then all you'll have left is this gigantic MHC1 negative tumor, and it'll just keep growing, and it won't be controlled by the T cells anymore. But there is like another immune cell that we have, braver. Oh my gosh, I think your name is Brave Traveler, right? I always read your name. I'm like, Brave something or other. Brave Traveler, welcome to the stream. Also, Avocado Man, hello there. Aw, that's fun. I just get super excited, okay? How are you, Brave? How is your week going? We're talking about how your immune system, like how different immune cells recognize cancer cells and how they kind of work together. So let's say that you have this loss of MHC1, you can't have your T cells killing the cancer cell anymore. Well, you do have another immune cell in your arsenal. That is a natural killer cell. Okay, we're just going to make it really big and very orange. I need to work on my drawing skills. Also, I think I need to charge my pen at some point. So we're going to say this is a natural killer cell. So natural killer cells, they are part of your innate immune system, which means that they don't necessarily need they don't need this whole T cell receptor and then peptide MHC interaction to actually get activated. They just get activated based off of a direct interaction with an inhibitory or an activating receptor. So NK cells normally, they survey your body looking for cells that don't have MHC1 because that indicates that maybe a virus or a cancer made the cell stop expressing MHC1. We're just going to kind of say that. So viruses actually have a different actual miRNA, so like different mini RNAs and other ways that they can literally shut down your cell's ability to make MHC1. And this protects the virus from getting killed because it gets to live inside of that cell now and continue replicating until it kills the cell, and then it bursts, and then it goes out and infects more cells. So NK cells, through like evolution, they've existed, or they've come to exist as an immune cell that recognizes cells that have been hijacked and no longer express MHC1. So normally your NK cell has a receptor that will interact with, like, say, so this one is going to be an activating receptor. And it will interact with different proteins that are induced kind of by stress. Like, let's say the cell is virus infected or it has cancer. The byproduct of having like a lot of DNA damage, for example, inside of the cell, is that this is going to cause a lot of like protein buildup, which will kind of activate certain signaling pathways inside of the cell, and they'll start expressing certain proteins, and these proteins, they're recognized by the NK cells specifically, and we just call them like NKG2D ligands, for example. So this receptor here, we're going to call that NKG2D. That's an activating receptor, for those of you who care. Otherwise, you can just think of it as like the green light. Like if there's any interaction with this receptor, it tells the NK cell to kill. And so then here's the little ligand for it. And this is induced by stress. And so that interaction without inhibition will allow the NK cell to kill the tumor cell. You're doing okay, brave traveler? I hope your week is going well. I'm doing pretty good. I'm doing good. Blind homeostasis hypothesis seems worth more research. I am not sure I've ever heard of this hypothesis, Free Nukes. I'm going to put this in my search history for later. Can you tell me a little bit about this? Let's see, blind T cell homeostasis. Oh, interesting. Okay. So it's a model. Okay, blah, blah, blah. So the blind T cell homeostasis hypothesis states that the total T cell population in the periphery, so like the entire body, is subject to regulation rather than regulation of the CD4 or CD8 subsets individually. So when they're saying regulation, do they mean regulation by T regulatory cells? This is based off of the original experiments by Adelman and Wolfsey on the effects of a CD4 T cell depletion in mice. So if you remove all the CD4 T cells. Okay. You know, this is very interesting. I wonder if I've heard about this in a different context. Let me look into this. I'm going to look into this off of stream. There's other related things like synaptic homeostasis hypothesis. Very interesting. So in terms of regulation, your CD4s and CD8s are regulated, well, your CD8s primarily are regulated by T regulatory cells. Oh, I see your explanation. Also, hang on, I'll read Space Turkey and Laura's comments in a second. Recently, it has been proposed that, oh gosh, why is my thing in the way? That a cell count is a normal cell count? I'm sorry, there's like a little icon covering up your message. Oh, recently it has been proposed that normal T cell counts are maintained by a homeostatic mechanism which is blind to the distinction between CD4 and CD8 T cells. Normal T cell count. Interest in this blind homeostasis hypothesis stems in part from its implications regarding the pathogenesis and treatment of HIV. In this report, BHH was tested in CD4 deficient mice. We found that as predicted by BHH, despite the absence of CD4 T cells, CD4 deficient mice maintain normal absolute T cell counts. So in the absence of CD4 T cells, the overall number of T cells, so I guess in this case it would be CD8 T cells, and perhaps other smaller T cell subsets like gamma delta T cells. They kind of proliferate to make up the full niche. So this is kind of something that we know happens. I didn't know that there was a specific hypothesis that was trying to... Yeah, I need to look into this. But basically, if you do remove a subset of T cells, you will get kind of this... basically, that the T cells that are pulled out will be replaced by whatever remaining T cell is there. And this is kind of relevant to different T cell clones. So in your body, you have many different types of so when I say a T cell clone, basically what I mean is that let's say this is a CD8 T cell, for example. So you'll have we're going to use different colors to represent each clone. And there's hundreds of thousands of different clones in the body, but the reason why they're unique is because they each have a different T cell receptor that recognizes a specific peptide MHC complex. And this allows your T cells to be able to recognize all different types of potential cancer proteins or different bacterial proteins. And so you get these clones of T cells. Now, usually of each individual type of clone of T cell, there's actually maybe about 1,000 or so cells. It might be slightly more. I think it might be 150,000 of each clone in the body. But let's say that one of the clones is activated. Like, let's say that this clone is activated. It recognizes its specific protein, which maybe is a certain type of viral protein. Like, let's say this T cell here, its T cell receptor will always recognize a protein from, let's say, I don't know, a different, like a specific type of cancer. We'll just say like a cancer antigen. And so when it recognizes that peptide, it'll make lots of copies of itself. And these copies will sort of overwhelm the total number of T cells in the body and will sort of, I guess, dominate as the most frequent T cell clone in the body. So if you were to like take all the T cells out of someone's body after they have this immune response, you would see like, oh, maybe 40% of T cells are this specific clone if you use an antibody that's specific to this T cell receptor. And then these clones will sort of become less, or they'll stay at their current number. What valid control methods exist to isolate and identify any virus? Any genome sequence for any claimed virus. To isolate and identify any virus. So normally if people are trying to identify a virus, usually it's helpful if this virus has already been sequenced, because then you can just go into like PubMed or NCBI, and just blast and look for the specific genome for that virus. I'm not sure what you mean by valid control methods. I guess, like, what if there was a situation where you had multiple viruses in a specific, co-infecting? Like, let's say you have 2 viruses co-infecting a host. How would you isolate those different viruses? Any genome sequence for any claimed virus. So generally, what you can do is you can isolate a singular like viral particle, and then you can have that proliferate in a lab setting, and then you can basically remove the DNA from that and then sequence that. Counts in the blood and spleen primarily through a marked increase in CD8 T cells. These findings provide strong new support for BHH, similar articles. Basically, they don't have a clue yet. Yeah, this is interesting, Freenukes. I'm gonna look into that some more. To this date, there has never been a complete sequence, though many continue to try. Still no valid control methods for any Any claimed virus. Okay. This is, yeah, so I I am not a virologist. Am I whispering? I'm not whispering. But yeah. So basically, if you know more about that, then you are the expert. Kaheni, welcome to the stream. How are you doing? I hope your week is going well. All right. So basically we have this. So let me just check. Hang on a second. Why is this window? Okay. I can hear you. Your music is a bit louder. Oh no, has it been loud this whole time? So this is what happened. I started up my stream, but I had reset my computer to update certain apps so they would work better. And unfortunately, it reset my audio. So I had to scramble to put that together. Yeah, fortunately in my VODs, I never have my music saved. So if there was any part that you couldn't hear, you could always hear it in the VOD. That's my backup. That's also why sometimes I'm not aware of the whole my voice to music input ratio, because I only hear the VOD without the music. So sometimes, unless I'm looking at the levels on my little sound control board, I can't really tell. Anyways, thank you for letting me know. All right. So honestly, I think this was actually sort of the end of what I had planned for today. Water to bean ratio. Yes, the water to bean ratio matters a lot. I'm curious about Free the Nuke's little blind homeostasis hypothesis. This will be something that I look into, and I will add it. I'm going to just add this to my little sheet, so I try to keep track of different questions that are brought up in chat on this little Excel sheet. I don't get to get through all of them, but it's here and it exists. And then, yeah, there we go. All right. But anyways, I do have a busy day tomorrow, so I just realized it was 11, so I'm going to be ending the stream soon. But for those of you who were here and hung out and listened to my random discussions, I appreciate you. Thank you. We are gonna find someone to raid. I am gonna check my list and see who's online. Oh no! Okay, I have a question. For those of you who normally watch edgy streamers, have you ever watched Homozygote? He's very interesting. He's very opinionated, but also interesting to talk to. But he's no longer streaming. I was gonna send you guys over there. Let's see. Okay. Well, okay, let me see if there's any other edgy streamers who are on. No, it is very late. So I'm going to send you guys over to one of my new streamer friends. His name is Reese Cups. He's a very chatty person. He's not doing any sort of science and technology right now, but he's always really cool to talk to. And he is currently playing Sekiro, which I believe is some sort of, I don't know, what is this game? Oh, actually, he's not even playing that. I don't know. Anyways, I'm going to send you guys over to him. He makes everyone feel welcome, which I think is very important. So I will let you guys hang out with him. And, Laura Space Turkey, Jandre, Hoju, Free the Nukes, Kenny, all of you guys, if I missed you, I'm still thinking of you. Thanks for hanging out. I hope you guys all have a fantastic rest of your week, and I will let you guys know in the Discord if I do end up doing a Bripe stream. It'll be an IRL stream, so you'll get to see some nature. It'll be outside of this little cave. And yeah, thank you for popping by, Laura Willingham. I appreciate that. Also, if you're a virologist, I might have some questions for you. Gear, thank you so much for hanging out and being an awesome mod. I appreciate you. You were, I think, one of the lone mods tonight. Scruff was here, but I think he went back to his VR movies. So thank you for that. I appreciate it. All right, so I'm going to send you over to Reese Cups. He's really funny and awesome and just, again, a wonderful, welcoming person. So give him a follow and say hello. And yeah, I will see you guys either next Tuesday or Saturday. Have a good week. And oh, yeah, you can spam coffee cups for him so he knows that the coffee rate is coming. There we go. If you're a subscriber, you can use little heart lattes. Otherwise, oh, I think it's going. Hang on. Let's come up with a little cute raid message. It's lots of heart lattes. I don't know. I'm done. All right, guys. Have a good rest of your week.